this is that it would seem to be as good as any other bacterial strain that you might pick it for addressing those questions. But we're also interested in the idea of using the phages to develop tools for mycobacterial genetics and the genetics of TB and the non-tuberculous mycobacteria, which were then genetically intractable. And as you can see from the phylogeny to the right here is Mycobacterium smegmatis, which is a relatively close relative of these other mycobacterial pathogens. We set about and sequenced the first Mycobacteriophage in L5, and that was reported in '93. But clearly, we needed to learn more about more phages and more genomes to do comparative genomics. And we helped to develop these integrated research education programs, of which the C-Phages program is perhaps the largest. Now with perhaps over 40,000 students in total, mostly in the US, have been through the C-Phages program, isolating new phages from the environment and sequencing and annotating their genomes. And we've learned that Mycobacteriophage diversity is impressive, substantial. It's at least 38 different distinctive types or different genomic types in terms of DNA sequence, just isolating phages from a single strain of Mycobacterium smegmatis. And there's substantial variation within each of those types or clusters, however you would like to define them. So this data originates from disseminated sources. So we consolidated in this database and website at phagesdb.org, where it's all publicly available. In total, students have isolated over 20,000 phages on hosts in the phylum of the Actinobacteria. We sequenced about 4,100 of those, most of which are in GenBank in the public databases. About 10,000 of the total phages were isolated on Mycobacterium smegmatis, and we've sequenced about 2,100 of those. So we have a high resolution of their genetic diversity, and those are the ones that we're going to focus on today. The diversity, as I said, is quite impressive. This is simply showing a gene content-based network phylogeny, showing one representative of each of the major groups. And you can see from this that there are many, many pairs of phages which essentially share no genes at all. And if you drill down into some of these clusters, this is a cluster F. For what it's worth, it's highly diverse. In this subcluster F1, you can easily identify a pair of phages when you compare them, which share about 40% of their genes overall. So substantial diversity all across this spectrum. Many, perhaps most Mycobacteriophages are temperate. If you just look at those major groups or clusters, more than 50% of them are temperate. So there are lots of lytic phages, but many, many temperate phages. And phages that are isolated that are lytic in those clusters, but also naturally what we would think of as naturally occurring clear plaque mutants, lytic phages, within clusters that are otherwise temperate and therefore have been derived naturally from temperate parents. These temperate phages can form prophages in lysogens that can be readily constructed. They either integrate into the genome or replicate extrachromosomally. And there's multiple different attachment sites spread around the chromosome used for these different phage integration systems. We've studied these integration systems in some detail. These phages use both tyrosine family integrases as well as serine family integrases, and there's non-canonical systems that depart from the lambda model for phage integration. For example, these integration-dependent immunity systems, which are unusual in that the ATP site for phage integration is located inside the gene for the repressor, such that in the viral form of the genome, the repressor is expressed. It has an SSRA-like degradation tag on the extreme C terminus, is degraded and is non-functional for superinfection immunity. But what happens is that when the phage integrates, the action of integrase, recombining between ATP and the host B site. So you now have an integrated prophage. This ends up separating the 5' and the 3' ends of the gene. So now this SSRA tag is removed, and you now form a stable form of the repressor, which confers superinfection immunity. Clearly an interesting twist on what we understand from the canonical phage integration systems. Just some general comments about the genomes. The phage genes are on average about 2/3 the size of bacterial genes. And so the phage genomes are replete with these small open reading frames, of which many are of unknown function. We think that many of these are implicated in the dynamic interaction between the phages and the hosts, either by conferring defense or exclusion in lytic growth, or acting as counter-defense or counter-exclusion proteins, although that really needs to be tested with many more of the hundreds of thousands of these genes of unknown function in these collections. The genomes are architecturally mosaic. They're constructed of genes, mosaic units, if you like, or tiles, often a single gene, which are related to each other, but in different genomic contexts. So over here, you can see an example of a couple of genes in 2 different genomic contexts. There's no DNA sequence similarity, and the flanking genes are distinct to the left and the right. And we can find many, many examples of these mosaic relationships. So this really explains the primary architectural construction of the phages. Likely being created by illegitimate recombination over long evolutionary periods. The phages have indeed turned out to be terrific tools for doing bacterial genetics and tuberculosis genetics, as well as phage genetics for delivery of transposons, reported genes, etc. Widely used plasmid vectors that use the integration systems to give single copy stable recombinants in these strains. Extra chromosomal origins of replication, non-antibiotic selectable markers based on phage immunity, and recombineering systems using the Mycobacteriophage recombinase proteins for mutagenesis of the bacteria and for engineering of the phages in these systems. I'll just comment briefly on this more recent system that we've described, which we think is useful and maybe a broadly applicable approach, which we call crispy bread or crispy brip, where what we're doing is doing a combination of the recombineering. So we co-electroporate genomic DNA with a synthetic substrate, for example, to create a deletion that goes into a recombineering strain. So you get proficient recombination between these. But then we can enrich for the progeny that we're after by counter selection against the parent phage using Cas9 CRISPR selection. So we make a plasmid expressing a guide RNA that targets the parent, as you can show here, and then you get very efficient recovery, and just by PCR, a very high proportion of the progeny are the desired recombinants. And so this can be done using electroporation to introduce the phage DNA. That's not always very efficient for some phages. And so it can be done by infection, where you simply infect the cells, recombineering cells, and then recover the progeny, select with the CRISPR counter selection. And this is particularly useful for constructing more complex recombinants of replacements, insertions, etc. So we've learned a lot about the mycobacteriophages, but had never really considered them in any serious sense for their therapeutic potential, although the mycobacterial infections are clearly challenging clinically, very challenging, and there's a clear need for new therapeutic approaches. And so phages offer some potential hope for that. Infections with non-tuberculous mycobacteria, like Mycobacterium abscessus, which is the one I'll focus on, are especially challenging. They're intrinsically antibiotic resistant, as well as having some acquired resistances. And patients are often treated with antibiotics for long periods of time with poor outcomes. And so these are long therapies over several years. The antibiotics are toxic and often not well tolerated. And there are few alternative opportunities for treating them. And so we think phages have some utility there. TB is a different kidney disease altogether, but we think there's also the potential for contributions for phages there. So I'm just going to go through some of the challenges here briefly, and then I'm going to show you some specific examples. So first of all, few phages have been isolated directly on any strain of Mycobacterium abscessus, and although we've tried quite hard, the yields are very poor, so quite a bit different from the experiences we have isolating Mycobacterium smegmatis phages, which is actually relatively straightforward. However, we know that some M. smegmatis phages, a subset of that diversity, have broader host range, and in fact, not only Mycobacterium tuberculosis, but also at least some of these Mycobacterium abscessus strains. However, we also know now that there's a great deal of variation, as I'll describe, in the phage infection profiles amongst different strains. So phages that work for one strain don't work for other strains. And that really there's rather a dearth of phages in general that infect the M. abscessus strains, and this is currently a limitation in thinking about a broader utility. And obviously many of these phages are temperate, and so if they're going to be used therapeutically, they need to be engineered so that they're not temperate, so that they kill efficiently and are lytic. And the M. abscessus strains are actually quite interesting because they're replete with prophages, which are likely involved in the phage infection profiles. And of course, we always worry about resistance being emerging during a therapy. So at the end of 2017, we were contacted by a colleague, James Soothill, at the Great Ormond Street Hospital in London. They had 2 pediatric patients with similar profiles. They both had cystic fibrosis. They'd both had double lung transplants, which was necessary in order to overcome the severely impaired lung functions. So both were on immunosuppressive drugs in order to protect the newly transplanted lungs, and then both had got these disseminated Mycobacterium abscessus infections, which were drug resistant, difficult to control. Both patients were sent home on palliative care with essentially no more options for treating the infections. And so one of the family members had inquired with James about the possibility of phage therapy that they'd read about online somewhere, and they sent us the strains with the hope that maybe we could screen them for phages. I thought the idea of using phages therapeutically was an unlikely option, but we were interested in questions such as host range and specificity, so we were happy to do this. Unfortunately, one of the patients died a couple of months or so after we received the strains, and we were unable to be of any help. The other patient, however, we did an extensive search for our phages, and after many months, were able to identify 3 phages, which were then given as a cocktail. We wanted to have multiple phages because of the concern about resistance. And these were delivered intravenously twice daily at a 10 to the 9 PFU per dose. That dosage is a guess because obviously there really isn't any good data that says what optimal dosage should be in these types of cases. And this was delivered over an extensive period of time, years. The phage is very well tolerated. There's no adverse reactions. And within 4 to 6 weeks, there was clearly good clinical outcomes and benefits. This infected node in the liver, which was very serious, appears to be nicely resolved. The skin nodules, which were pervasive on the limbs, mostly cleared up. And the sternal wound, where the transplant had been done, which had failed to previously heal and to close, was now closing up quite nicely. We were pleased that we didn't see any evidence of phage resistance in this case, and we didn't see any neutralization of the phages by the patient, by the immune system, but because the patient was immunosuppressed, that was also not anticipated. So here is Isabel when she was doing well. She since passed away early this year, sadly, which also reflects the complicated nature of many of these cases that we've been involved in. There's lots of other conditions to be considered clinically. And this was written up in 2019. And then since then, we've had a lot of requests from physicians with patients in similar circumstances. And so these are These are not as common as some of the other infections, serious bacterial infections, but when they occur, they're a total nightmare for the physicians, which is the reason why we've had all these inquiries from folks across the globe. About one every 2 or 3 days, we get an inquiry, and we respond by asking for the physicians to have their clinical microlab send us the strain. Most of these are Mycobacterium abscessus strains, and the others are a variety of other mycobacterial strains. And we then receive those strains, we streak them out, we grow them up, we test them, we use those plaque assays that you're familiar with, and look for phages that not only efficiently infect the strains, using a fairly standard, a more constrained set of standard phages that we think are the best candidates. And look for those that can kill the strains efficiently in the lab, in the hope that they will be the best candidates for moving forward with a potential therapeutic application. About 40% of the strains that we receive have these smooth colony morphotypes. M. abscessus comes in both smooth and rough colony morphotypes. Unfortunately, we don't have any phages that efficiently infect and kill any of these smooth colony isolates. So that is a major impediment to broadening this type of application. We can find one or more phages for about 75% of the rough colony isolates that we receive. And so we've then identified suitable phages, and then in about 33 patients, we've prepared and purified those in the lab, provided them for compassionate use under regulatory appropriate regulatory control. In the US, that would be using the FDI EIND process. And we've written up a consecutive case series of 20 of these patients. And also I'll make some comments about those and give a couple of examples. So these are complicated cases, as I said, and the administrations are not administered on an emergency basis. From a timing point of view, the patients have had these infections for relatively long periods of time. They've often been on antibiotics for a couple of years. And so there is not a severe time pressure in order to implement these, but also the phage applications need to happen over long periods of time, months or years. So these are not simple therapeutic interventions, although there's clearly a very important and a strong need for them. For those 20 cases, we have 11 out of those. I think there's fairly well-defined favorable outcomes, either clinically or microbiologically. 5 are kind of unclear, uninterpretable. If a patient, for example, died very shortly after administering the phage, and so you have no opportunity to really see if there's a good outcome or not. In 4 out of the 20, we see no obvious benefit from the phage administrations at all. We don't know why, but we would clearly like to understand that. Interestingly and helpfully, I think, in 11 patients, we administered just a single phage because that was all that we had available for those particular strains. And we've not seen any evidence of resistance in those infections. So there's no, we don't find resistance, and we don't see treatment failure due to resistance. And this is consistent with what seems to us to be a very low incidence of resistance in vitro. If we just do the infections in the lab with 10 to the 8 or more bacteria, it's actually quite challenging to identify resistant derivatives, although we have managed to isolate and characterize some. We should not be surprised that in patients that are immunocompetent, they generate neutralizing antibodies, probably starting sort of 2 weeks and onwards. In some cases, there appears as though that may be limiting for the efficacy of the phages therapeutically, but in others it isn't. And so the impact of the neutralization, at least for these patients, is not clear. And as I mentioned, there's great variation in phage susceptibilities from strain to strain. So these therapies have to be personalized, and going beyond personalization to broad usage is a major challenge for which there are no clear resolutions currently. So I'm just going to show 2 of these cases very briefly. This is a middle-aged male patient with a disseminated infection with Mycobacterium chelonae, which has caused these quite extensive skin nodules or abscesses on the limbs, as you can see here and in the scan and here. It's quite extensive. We identified only a single phage, which was muddy, one of our favorites actually, and this was administered after the failure of all of these antibiotics that have been administered here. It was treated for IV twice daily for about 6 months, and over that period there was actually very nice resolution of the infection, as you can see in these scans. And biopsies after 6 months, the biopsies have failed to produce any Mycobacterium chelonae strains that could be grown out. This patient mounted quite robust antibodies against the phage that were potently neutralizing, but it doesn't appear to have impacted the outcome of the infection. The second patient is a little bit different and in some ways a lot more complicated. This was a young adult male, cystic fibrosis, is immunocompetent, but has this highly drug-resistant pulmonary Mycobacterium abscessus infection. It'd been in and out of hospital for long periods of time, as I'll show. And we were able to identify 2 phages that infected that particular strain. And that was administered IV for a period of about 12 months. Now, for this patient, and this is relatively common, his Mycobacterium abscessus infection was considered as a negative aspect for consideration for lung transplant. So his lung function was sufficiently poor that he really needed a transplant, but couldn't get it because of the M. abscessus infection. And so after a year of treatment with the phage, he regained transplant eligibility because of clearance of the phage of the bacteria from sputum, received a transplant and is doing very well, is now off antibiotics, is off phages, and is just having to manage the rejection for the immune rejection of the lungs. And again, we saw no phage resistance and some potent neutralizing antibodies for one phage, but not for the other. So this is just the patient profile here. You can see this is the number of days, and so this is over a significant number of years. All of these hospitalizations because of the CF and those complications. You can see his lung function here, which is just declining, and this is actually a very poor level of lung function down here. The red bars just show when they started culturing Mycobacterium abscessus from sputum. So this happened sort of repeatedly. The phage were initiated here after he had had all of this long, complicated drug therapies. The antibiotics were continued, as is all of these cases. The phages were added, and then it took a couple of months in order to get culture negative sputum, but then that state persisted with one only very weakly positive culture here, and then this is where he received the transplant and regained his full lung function. This was a helpful case in part because there was a series of biomarkers that could be used to follow during the course of the infection, which tells us a little bit how the patient has responded.
Graham, could I maybe just interrupt you, sorry there, to go to maybe your last slide and draw your conclusions?
Yeah, this is a 30-minute talk though, and I've talked for 21 minutes. So if I can just I'll move along quickly, and I'll be done in 25 minutes. Okay. So as I mentioned, the engineering the temperate phages to be therapeutically useful, and this is an example of a phage called Fionnbharth. We simply made a substrate and have engineered out this central immunity cassette in order to make this nice clean deletion. It's a non-recombinant but engineered phage that can be used therapeutically. As I mentioned, for all of these strains, we do test that the phages can infect these strains efficiently and kill them. So this is an assay with one of those strains with about 2 × 10⁸ bacteria. It's killed with muddy and liquid infection, and there's no survivors at this level. So this is an example of this low incidence of resistance. So I just want to make the point that this is a complicated slide, but that even though these strains phylogenetically, some of them are quite closely related, the phage infection profiles vary enormously. So we can understand this in the context that even though these strains are CRISPR-free, the Mycobacteriophages don't have CRISPR, they do carry many of the defense systems that we heard Frank Nebrega talk about earlier. They also have lots of plasmids, as is shown over here, but they're also rich in these prophages. And we think these prophages are playing large roles or substantial contributions to the phage infection profiles. We've, because we're having to identify many of these prophages, we have some software that we've just published which facilitates this, and we can pull out these genomes with some precision for the AT-L and AT-R junctions, annotate these, and do the comparative genomics just as we would for the other smegmatis phages. Nucleotide sequence-wise, these are all distinct from the smeg phages, but we can analyze them in similar ways. We've been interested then in asking whether we can get spontaneous induction of these prophages from some of these strains and then identify other strains that they can infect. So we're doing essentially a very large number of pairwise crosses looking for a strain that releases a phage and a strain on which it plaques. And this is an example of that screen. So we've identified quite a lot of these now. We can sequence them, do all the genomics, we can look at the morphotypes in the EM, and then we can test each of these back against our large collection of strains. And in fact, we can identify several that now have new tropisms. In other words, one of these induced prophages, or the now a lytic phage, that can infect a strain for which we don't have any other phages, and that's an example. Of course, these are all expected to be temperate, so they need to be engineered before they can be used therapeutically, and that has to be done in Mycobacterium smegmatis, because in Mycobacterium abscessus, because they don't grow in Mycobacterium abscessus, and so there's that engineering challenge which we are currently trying to meet. So let me just summarize very briefly. I think that there's substantial therapeutic potential for these phages for these patients who really have no other options. Personalization is absolutely required at the moment in order to move forward with this. But hopefully, if we can understand and tackle the host range and the specificity issue, we can break this out into a more broader utility. Many thanks to a lot of people that have been involved in the work that I described, especially folks in my lab here. And I want to just point out to Becca Dedrick, who has spearheaded this effort. Here's Becca, who's spearheaded the therapeutic aspects in the phage, with other major contributions by folks in the picture. And I'll be happy to answer any questions. Thank you.
So that was a wonderful, fascinating talk on personalized medicine and opportunities with phage research. Any questions from the audience? Can I ask one, Graham, with regard to the neutralizing antibodies that you saw generated in some of the immunocompetent patients? Have you started epitope mapping any of them to see which particular areas of the bacteriophage seem to be more prone to, we'll say, antibody-mediated neutralization?
No, that's a great question. We would like to. What we know, however, is that there is clearly a distinction between seeing a response that you can measure by ELISA and seeing neutralization. So in some cases, patients have an antibody response to the phages even prior to therapy. So they have seen a phage or a phage motifs that are like that before. It's just that they're not neutralizing. So that's presumably a difference between having specificity towards epitopes that are involved in receptor recognition and binding as opposed to elsewhere on the capsid. We've looked at some specificity to phage proteins by Westerns, but we haven't mapped any of the epitopes specifically. And the challenge is clearly dissecting out those interactions which are neutralizing and those that are not. And I think that's an important priority.
Yeah, correct. Absolutely. Any questions from the floor? Yes, move on. You might just identify yourself for...
Yeah. So I'm Dibyendra. I'm a postdoc at University of Exeter. In all of your cases, I guess, because the treatment involved continued antibiotics as well as the phage. So have you also looked at the phage and antibiotics together in the lab? Does that have any difference in the outcomes?
So first of all, yes, we have looked at some of those interactions in the lab. I mean, I think that it's not surprising if one would see synergistic reactions, but it also wouldn't surprise me if you see just additive or even antagonistic reactions, depending on what the antibiotic is. And so we do sinograms and things to try to understand that. We have a lot of strains to characterize in that way, as well as many different antibiotics. And so the interactions are complex. And it also really matters whether the strains have this rough or smooth phenotype, as we're discovering as well. What we don't see is any obvious correlation with which patients have which antibiotics and the phages, or whether antagonism is a plausible cause for loss of or lack of efficacy in the 4 patients that we saw. We don't think it is, but we can't rule it out, and we would really like to understand that question. And it's clearly a general question in all of these types of therapies, and I think it's an important one.
We have another question here from the
follow. Sorry. Because these phages are mostly temperate, so does the presence of antibiotic affect life cycles usually, choices? Do you have any ideas on that?
So we don't, but let me just emphasize that the phages are temperate, but we don't use them as temperate phages. We engineer them so that we make them lytic by removal of the lysogenization genes, the repressor especially, but we also like to take out the integrase as well. And so they are these engineered phages are lytic, and we can show that they very efficiently kill the bacteria in the lab. And so we don't have to worry about an interplay with the antibiotics or anything else for that matter that influences those life cycle decisions.
Great stuff. One last question, if it's quick.
Hello, very nice talk. My name is Ville Freeman from the University of York. I was interested in the lack of resistant evolution in vivo. Do you see rapid resistance in the lab in vitro against the phages, and is there any sort of costs involved? So is it sort of costly, so it's not selected in these in vivo environments?
Yeah, so just briefly, I tried to make the point, but maybe it was brief and unclear, that resistance in the lab is also at a very low frequency. It's not that it doesn't happen. We have it. We have genes with mutations in them. They are not cell wall genes as far as we can tell. So we don't understand the mechanisms per se. Whether there's a cost for pathogenicity and virulence and growth in vivo is an important question that we'd also like to address. It's important to establish good animal model systems for doing that. We're not doing that, but other folks are, and they has to be done in these strain-specific ways. But once we have that, it should be relatively simple to ask whether any of the phage-resistant mutants are really compromised for their growth in vivo. I suspect that they will be, but it'll be interesting to find out.
Thank you. Great. Thank you, Graham, for those comprehensive answers. Okay.